close your eyes. Okay, good. Okay, orbicularis oculi. This muscle was not on your list, but just to give you an idea of what the significance is of this muscle for lecture next week. Levator palpebrae, elevate palpebrae. Palpebrae is the technical term for your eyelid. So if you're going to elevate your eyelid, this is going to open your eyelids. That's the job of that muscle, okay? And so this is our lateral view. One thing that we did not go over in lab that you don't need to know for this week, what you need to know for next week is know the pathway of a tear. This is what's called the lacrimal apparatus. Where our tears actually end up draining, they end up draining here. But where our tears are actually produced are at the lacrimal glands that are actually all the way up here. So the way that this works is that these lacrimal glands, which are exocrine, they're exocrine glands, so they are not ductless, they are going to produce tears, and then the direction of the tears are going to go medially and inferiorly. And so it's going to sweep across the anterior surface of your eye, and so it's going to be in this order. We start off with the lacrimal gland. It produces tears. Then it's going to drain into the tiny little opening that is there. That next time if you ever look at it in the mirror, look really close and pull this down, and you're going to notice there's a tiny hole that's there if you haven't noticed it already. That tiny hole has a name. It's called the lacrimal puncta. And then from the lacrimal puncta, it's actually going to drain into a very tiny canal. This little tiny canal is called the lacrimal canaliculi, tiny canal. That drains into a much larger area, which is called the lacrimal sac, and that will lead lastly to the nasolacrimal duct. So this is why when you've ever had a really good cry, your nose is going to start to run right after. It's because it's not just the mucus that is coming from here, but it's mixed in also with all of the tears that would have come from here and swept in and drained and come out that way. So that is a lacrimal apparatus. Extrinsic eye muscles, our cranial nerves, LR6, SO4, all others 3, just remember that. And I think with this we can kind of go past this, because this is just kind of as a review for you that I'm pretty sure that you were studying this, you know, last week in order for you to prepare for this week. And so again, I'm not going to stay too long with it. I'm going to skip some of it because all of it we've already seen, okay? Aqueous humor and vitreous humor, we're familiar with those. Just to let you know with your aqueous humor, this aqueous humor, all of our humors generally are pulled from the bloodstream and they typically are going to be returned to the bloodstream. So one of the places where we actually try to return this humor to the bloodstream is through a little opening called the scleral venous sinus. And so take a look at where we are with the eye. This is our cornea, of course. This is our iris. That opening is going to be our pupil. The scleral venous sinus is really at the junction between the cornea and the sclera. So what is the significance of that scleral venous sinus? If for whatever reason the scleral venous sinus is not accessible or it is plugged for whatever reason, what will happen is that the aqueous humor is not going to be able to drain into there. And when it's not able to drain into there, it starts to accumulate. And once the aqueous humor starts to accumulate, that's also going to affect the shape of the eyeball and the pressure within that eyeball. How many of you have to wear corrective lenses? How many of you have ever gone to the optometrist and after that they blow a little puff of air into your eye? Okay, do you know what that's for? If you ever asked, or they would have told you, why do they do that? What are they looking for? It's a disorder of the eye, and it starts with a G. Glaucoma. Okay, what they're doing is, so one of the manifestations of glaucoma is that your aqueous humor has accumulated. And when it's accumulated and it doesn't drain, it's going to make your eye a little bit more spherical, and then the pressure within it, the intraocular pressure, is going to be greater. When they introduce a puff of air, what they are measuring and they're looking for is if in response to that puff of air, your eyeball should be able to bounce back to a certain degree. If it cannot bounce back to a certain degree, that means that it is so full of aqueous humor, and that's what's going to affect it. So that's why that is one of the well-known tests for glaucoma. They want to see the intraocular pressure within your eyeball, because it should depress and bounce back. If it is too full of fluid and the pressure in there is too high, it's not going to be able to bounce back in the way that it should. Okay? So that's the significance of the scleral venous sinus in terms of our ability to drain that extra aqueous humor and return it to the bloodstream. And so it is constantly renewable. So the next parts are ones that you would have seen. We've got our fibrous tunic, of course. We've got our vascular tunic. Remember that these two areas are made up of a particular type of muscle. What is the type of muscle that they share? What kind of muscle is the iris and ciliary body made out of? Here are your choices: skeletal, smooth, and cardiac. What kind of muscle are they made out of? Can't be cardiac, right? Smooth. If it were skeletal, then that means that we would have to voluntarily move our iris all the time. And thankfully, we don't have to do that, right? So both of them are made of smooth muscle. The 6 extrinsic eye muscle, what kind of muscle is that made out of? The 6 extrinsic. What is this made out of, everybody? Because we can do that voluntarily. Skeletal muscle, very good. So we have our extrinsic muscles are made up of skeletal muscle, and our intrinsic ones, which are these are considered the intrinsic muscles, they are made up of smooth muscle. The last tunic here, of course, is that retina. There is a border between the ciliary body and the retina. Like I mentioned last week, the retina is not going to cover the entire anterior portion of the inside of that eyeball. It's only going to be kind of a little bit more than half, maybe about 2/3. And so there is a border between them. It's called the ora serrata. There are 2 layers of the retina. Pigmented, we don't talk much about. The pigmented part is the one that when somebody has a detached retina, this is where the detachment is going to happen. It's going to happen at the pigmented layer. But we didn't talk much about that. We talked mostly about the neural layer. The significance of the neural layer is that we have 3 cells. And so from vitreous humor, or posterior cavity, all the way to what is closest to the choroid is the following: ganglion cells, bipolar neurons, or bipolar cells, and then the photoreceptors. And so that's exactly what is here. And let me see if I've got, no, I don't. But in your histology, what I would like you to know in that histology, what is in the guided lab lecture is knowing which cells are which. What is your clue? Look for the choroid. If you find the darkly colored choroid, the cell layer that is closest to that choroid has to automatically be your photoreceptors. And it's going to stain dark. If you have a group of cells that are going to be facing what looks like an empty space, then you're likely looking at the ganglion cells. And then this is the one that would be sandwiched in between. So here is our ganglion cells, our bipolar cells, our photoreceptors. This is your pigmented layer, and this is where your choroid and your sclera are expected to be. So just to give you that idea. Now, this is one part that is, or can be, a little bit confusing. So with that, I'm going to go to this. I said to bring this one this week. We're going to go over some of it, at least in the parts that we didn't get to go over in lab. We kind of already answered this, everybody, so you should be filling this out already. The pathway, visual pathway to the brain. So this is asking for number the following steps in order to describe the pathway that light would have to travel through our eye to be picked up by photoreceptors still in the brain. Okay. You're going to notice that some of the steps here, pay close attention to the part about the bipolar cells and the ganglion cell bodies. Because they start with the term passes. Passes. Okay, so when we look at that, I'm going to put it side by side. arrows. One is light in, the other one is signal out. So describe to me, what do you think is happening over here? Tell me the order that light is going to travel when it enters your eye. What is the very first structure that light is going to enter? This is preparation for next week, guys. Cornea, very good. Okay, after the cornea, where are we going next? The pupil, excellent. Which is controlled by the iris. Okay, what's behind that pupil immediately? The lens, cool. Okay, once we go past that lens, what cavity are we in right now? Posterior or anterior? Once we go past the lens? Past the lens. Posterior. Okay, so we're going through that posterior cavity. Now there's the part where we actually hit the retina. This is the direction that light has to go through. It has to actually go between these first two layers of cells and then get to the photoreceptors where it will be converted into a neural signal. So just to let you know, these ganglion cells and bipolar cells, remember, they are neurons. Neurons don't care anything about light itself. They don't know anything about light. They don't know what to do with light. They are not able to pick up on any light because they are neurons. What is the language that they understand? Action potentials. That's all they understand. All they understand are action potentials. When light is coming in, light is not coming in as an action potential. It's light. So what the light has to do, it actually has to go in between those cells. And once it goes in between those cells, then it gets picked up by the photoreceptors. And this is where we're going to convert that light into something meaningful, which is a neural signal. And so then the neural signal ends up going in reverse. We have the neural signal being generated here because of the photoreceptor. And then now it's going to go through the bipolar cells. Now it's going to go through the ganglion cells. And then we are going to exit out the ganglion cell axons, which are going to lead us to where? The ganglion cell axons all join together and they become the what? Which one? The optic nerve. The optic nerve. Does everybody see that? Okay, this part's super, super important for next week, okay? Really important. So now that we've got our sensory tuning right here, we've understood our retina. That takes us to the specialized areas of the retina. These two areas are complete opposites of each other. Let's go over what we might know already. What is the significance of the fovea centralis? The macula lutea and the fovea centralis are related to each other in terms of your macula lutea and fovea, if you think of it as a target. The whole target is called the macula lutea. The bullseye is the fovea centralis. What is the significance of the fovea centralis? There's a high number of villi in this area. This is where you have the highest number of villi. Of which ones? I want you guys to think about it before you answer. Photoreceptors. Okay, photoreceptors. So what we can have is we're going to have our cornea and our lens. The job of the cornea and the lens is to bend light. And they have to bend it in a way so all of those light rays are going to converge as much as possible on that one spot called the fovea centralis. Because we have a high concentration, the highest concentration of photoreceptors at the fovea centralis, this is what allows for us to see clearly and when we focus on just one point. That's the fovea centralis. Give you an idea where fovea centralis is. If you were to look at a retinal scan, the fovea centralis is typically just going to be a little bit of ways from where the optic nerve is going to exit. And then speaking of the optic nerve, what is the other specialized area where that optic nerve is going to be exiting? That is called the what? The optic disc or the blind spot. Why did I say that this is the exact opposite of the macula lutea and the fovea centralis? How would you describe that then? I want you to take a moment and think about it. Why is it the exact opposite of the fovea centralis? Venture a guess, venture a guess. Right or wrong, it's okay. I just want you guys to know your voice here. Yes?
When all the light goes separated,
Good. Okay, so one of them all the light goes to because you have the highest concentration of photoreceptors. Okay. Describe then about the photoreceptor population at the optic disc / blind spot. You have an answer? Just tell me. The fovea centralis has the highest concentration of photoreceptors. What can you tell me about the optic disc? It's the exact opposite. It doesn't have any. It has none. Good. Okay, good. Good. I want you guys to find your voice. Find your voice. All right. So the optic disc, what makes it the opposite is that there are no photoreceptors. If your whole area is crowded with a whole bunch of axons exiting, you can't put photoreceptors there. That's why. So if light is projected and centered in the optic disc, that's an image that you can't see. That's why it's called the blind spot. Okay? So those are two specialized areas of the retina. In our dissections, we were able to see where the optic disc is, but we couldn't see where the macula lutea would have been because there's no core marking of it. In some of the models, they are going to show both of them. How do you know which is which if you have two designated areas? The cranial nerve number 2 has to be directly behind which one? The macula lutea or the optic disc? Everyone. Optic disc. Optic disc. It has to be directly behind the optic disc. Okay, so how do we see? How do we see ultimately? If you cover one of your eyes, you can still see both views, both fields of view. If you cover the other eye, you can also see both. So that means that you don't have a very clear dividing line in between. So you're going to have the visual field, this is the left visual field, the right visual field. You are able to get information from both of those visual fields, even if you have one eye that is going to be covered. And so your left and right is represented by the green and the purple respectively. So as you can see, we cover the side, we still have a purple, and we still have a green. And then same with this one, we still have a purple, and we have a green. What is the significance of this? Information about both visual fields, right and left, are going to come from both eyes. They are going to converge, and then they are going to cross. At this crossing, that's what we call the optic chiasm. And once we get past that optic chiasm, now we are going to separate information from their own respective fields, and they are going to be registered on the other sides of the brain. Okay? So your right visual field, all that information is going to end up in the left side of your brain. Everything from your left visual field will end up in the right side of the brain. But before we get to that optic chiasm, it's going to be shared between both. And then finally, where are all of our visual impulses going to end up? Where is our primary sensory cortex for vision? Is always the, everybody, what is this lobe? Everyone.
The occipital lobe.
What is this lobe? Occipital lobe. Occipital lobe, all the way in the back of your brain. Okay? And so this is just kind of describing the same thing, that you are going to get the retina from each one taking visual stimuli from both fields, and then where they finally cross and separate is off that optic chiasm. So just as a really quick review, now that we've got the path of the signal. So when we look at the path of the signal, we have to start with the cell where the signal was first produced. And that would be our photoreceptors. And so from here on out, this is the path of the neural signal that will result. Photoreceptor cell, then through the bipolar cell, through the ganglion cell, through the optic nerve, chiasm, the tract, the thalamus, and then finally the occipital lobe. So if there's any kind of defect in any part of this signal, or there's any kind of defect in the pathway of light before we get to the photoreceptors, that can result in partial or total blindness, some kind of a visual defect. So your concept check, I'm gonna leave this with you for your concept check, because this is something to kind of think about as you are studying. And in the meantime, let's go ahead and take a break, and then we are going to go into the ear, okay? The ear, and then have your lab worksheet ready for some of the parts that may be a little bit fuzzy, okay? Let's take a break. Let's take about a 5-minute break, a 5-minute mental break, and then we are